Okay, we're going to be discussing diffusion and osmosis. So diffusion is the net movement of solutes or particles from a region of greater concentration to a region of lesser concentration. So here in these beakers, or sorry, in these flasks, you will see that we have green food coloring that we're dropping in. So this area right here, where I've circled in red, that is the area of greater concentration. And so the particles, the blue food coloring, are going to move from this area outward. So you can see it progressing here as the particles are moving out from that initial area of higher concentration. And again, they continue to move out until eventually this whole beaker is equalized and there is no area of greater or lesser concentration. It's all equal concentration. So again, diffusion is the movement of solutes or particles from a region of greater concentration to a region of lesser concentration. Here's another picture. So if you think about this picture as you're drinking hot tea, and we'll think about this little drop here as being a sugar cube, and you're going to drop that sugar cube into your hot tea. So your solutes is this little sugar cube up here. So you drop it in this area that I'm circling in red here. That is your area of greater concentration. So your particles are going to be moving outward to try to equalize the concentration throughout this liquid. And you'll see that here. It's progressing from left to right. So here you see some movement of those particles outward until finally you get in this fourth beaker here where you see all the concentrations are equal throughout the solution. So diffusion and osmosis, what's the difference? So again, diffusion is the movement of solute or particles. Osmosis is the same thing except it's the movement of water. So again, diffusion is the movement of solutes from an area of high concentration to an area of lower concentration. So our solutes here, you can think about these solutes as being little salt crystals. Each one of these little circles here represents a solute, or for our example here, salt crystals. So when you have a high amount of salt here, it's going to want to move to areas of lesser salt over here to try to equal out the solution throughout. Osmosis is the movement of water from higher concentration to an area of lower concentration. So here, you see these black dots are going to be our water molecules. So you'll see on the right side of this image, we have a lot more water than we do on the left side. So you're going to have more movement or net movement of water from the right side into the left side until it equalizes the concentration of the water. And you'll see that here on the bottom picture, where now the water has been equalized and there's equal movement of water back and forth, because the particles in solution are not still. They're constantly moving around, but you're wanting to know what the net movement is. So it's almost like a subtraction problem. If you have four particles moving left and 6 particles moving right, then your overall movement is to the right. You have more particles moving that direction. So net movement is what we're concerned with here. And moving on to the next picture, here we're looking at osmosis in the cell. So the movement of water from an area of higher concentration to an area of lower concentration. This winds up being extremely important when you are looking at the human body. When you go in and you get IV fluids, they have to put in isotonic solution into that IV bag. Isotonic, you'll see that here, the middle one. Isotonic means that it's not a greater concentration inside or outside of the cell. The concentrations of water inside and outside are equal. Therefore, there's no net movement of water in or out of that cell. That's extremely important in the human body, because if we just put in straight water in that IV solution, then our concentration of water outside the cell is going to be greater than the concentration of water inside the cell. And water will continue to move inside that cell until it lyses or explodes the cell. And as you can imagine, if you lyse your red blood cells in your blood, you're going to fall into just a wide array of problems stemming from that. So you have to make sure that your solution is isotonic, meaning that you're not dehydrating your cells, or taking water out, but you're also not over expanding yourself by letting too much water go into the cell. So it has to be that iso. Iso means equal. Okay, so now let's talk about hypertonic and hypotonic. Hyper means a lot, hypo means a little or lower. So in this first picture here, what we have is the concentration of water is greater inside the cell, which means you have more solutes outside the cell, and your water follows your solutes. So if your solutes are greater outside the cell, it's going to follow those solutes. And this can result in cell shriveling or crenation. This last example over here is your hypotonic. If you're putting your blood cells into a hypotonic solution, that means that your concentration of water is greater outside than inside, and the water is going to move into the cell, which can, you can see right here, can cause the cell to burst. Okay, so in this image here, you're looking at osmosis, again, the movement of water. So you'll see your water out here, and these bluish purple dots are going to be your solute concentration, or your, for instance, salts. So your water is going to follow where there's more salt. So the water is going to go from the left side into the right side to balance it out. So your direction of movement here would be from your left to right. So it would be through this semi-permeable membrane, which is just a membrane that water can pass through. And then it will go up to this side over here, because it's trying to balance out that water and solute concentration on both sides. Okay, now we're going to be working with something. This is going to be similar to what you're going to see on a practical. Okay, so if we're given this, and we're not going to tell you whether it's isotonic, hypotonic, or hypertonic, okay, but you would see this beaker right here, and you see your key over here. So the dots that are filled in with black are going to be your water, and the open circles are going to be your salt. And in this, you can see that it says your concentration of salt inside the cell is 50%, and your concentration of salt outside the cell is 50%. So they're equal. So this would be isotonic. So it's asking which direction would the water move. So in this one, you can put a little equal sign right here. That means that there is equal movement inside and outside of the cell, meaning that there's no net movement across that membrane. Now the next one down here, again, we're not going to be telling you whether it's hypotonic or hypertonic, but here it says there's 50% salt concentration inside the cell and 25% salt concentration outside the cell. So remember, your water is going to follow your salt. So where's more of your salt? Your salt is inside this cell. So think about when you eat a bag of salty potato chips, the next thing you have to do after eating that salt is you have to chase it with some kind of liquid. So your liquid, or your water here, is going to chase the solutes or chase the salt. So being the salt concentration inside the cell is greater, then your water will be going into the cell, and you would draw little arrows showing that it's going to go into the cell. Okay, now let's erase this and go down to the last example here. Okay, the last example here is of a hypertonic solution. Let me get my little pen mark up here. Okay, in this one, we're told that there is a 50% salt concentration inside the cell and a 75% concentration outside the cell. So think of where the most salt is. The most salt is out here, the outside of the cell. And remember, your water is going to chase the salt. So your water is going to be moving outside the cell. And again, on our practical, you would draw the arrows to indicate the movement of the water. So again, let's see. So again, you want to make sure that you can draw these arrows on a practical. And that is it for osmosis and diffusion.